So in this lesson, I want to look at the pathogens part of communicable disease. So what is a pathogen? Well, a pathogen is an organism or virus which causes a communicable disease. So communicable means that it can be spread, and disease is talking about a disruption of your normal body function. So it's important to note that there are different types of pathogens that you have to know. It could be a viral pathogen, it could be bacterial, it could be fungal, or it could be a protista. Now, this is a protoctista, but we now shorten it down to protista. Now, these pathogens can either damage your tissue directly by using the metabolism of the cells that they're going to destroy. So if they're going to damage the host organism by using their metabolism, we're talking about hijacking the organelles inside to replicate themselves and then bursting out of the cell. Or it could be that they actually are going to release enzymes that will digest the cells, and then they'll use those cell components to actually utilize the reproduction process for themselves. So this is the direct way in which they can damage tissue. Now, an indirect way in which they can damage the host organism is by producing toxins, and those toxins will then have an effect on the cells within the organism. So there are specific ones that you have to know. So let's start with our viruses. So the viruses are non-living infectious agents. Now they are non-living because they do not have the necessary components to reproduce on their own. So therefore, what they do have is genetic material, which can either be DNA or it could be RNA. So they could be looking at whether or not you understand what happens if it's DNA or what happens if it's RNA. Now, the genetic material is always surrounded by proteins. So it's important to note that these viruses are going to invade living cells, and they can do that through a number of different ways. They can inject the genetic material into the host cell. They can be engulfed where they're taken into the cell, or what they can do is actually fuse with the plasma membrane on the cell. So it's important to note that there are different ways in which they can invade living cells, and this is dependent on the type of virus that is causing the infection. So what they do once they're inside is they want to take over their biochemistry, and they want to reproduce rapidly. So it could be that they're going to inject their DNA, and that DNA can then be transcribed and then translated, or it could be that they put their RNA, and that RNA can either be translated, or what they might do is they might end up converting that RNA into DNA. One of the interesting things about viruses is that when they have reproduced, what they're going to do is leave the cell by either lysis, which is where they're going to burst open the cell, or they can do what's known as budding, and that's where they take a portion of the membrane which forms an envelope around them. And this is what allows them to actually do this fusion with plasma membranes. If they have a portion of a membrane surrounding them as an envelope, that is therefore going to be the same biological membrane as the plasma membrane, allowing for that fusion to occur. So we also have some viruses which attack bacteria. Now, they are called bacteriophages, and it's important for us to understand that these viruses, whatever the virus is, because they are going to be using a host cell and they are going to be harming that host cell, they are the ultimate parasite. And it's important that you know specific examples. So the human immunodeficiency virus is one you need to know for your exam. Now, they are going to infect humans and some primates. Now, if they're going to give you information about the human immunodeficiency virus, they're expecting you to apply that. So it's important that you note that they target T helper cells. So this is where they could ask you to apply your knowledge to the immune system. So if they're going to target our T helper cells and reduce the numbers of T helper cells, they're going to affect the immune response to other pathogens. The human immunodeficiency virus actually contains RNA, but not only does it have RNA, it also has other enzymes present. Now, one of the enzymes you need to know about is called reverse transcriptase, and you'll learn more about that in Module 6 when you're looking at manipulating genomes. But it's important to note that the HIV has this RNA and an enzyme that can do reverse transcription. So what we're talking about is an enzyme that's going to turn our single strand of RNA into what's known as a complementary double-stranded DNA. And what can then happen is that complementary DNA can then be integrated into the DNA of the host cell. So how is it transmitted? Well, the transmission is through bodily fluids. Now, this could be through unprotected sex. It could be through inoculation of sharing needles. It could be contaminated blood transfusions, or it could be from mother to baby through the placenta. So it's important that you note that it is transmitted through different ways, and therefore they could again expect you to have that application question. So if it gets into the bodily fluids, it will then be able to be transported until it reaches its target cells, which are the T helper cells. The treatment side of things is actually the use of an antiretroviral drug. Now, the antiretroviral drugs, they could give you any example, but what they will be doing is if they give you information about a treatment, they'll be looking for you to apply your knowledge. So if they give you information about a treatment which is an antiretroviral, which is an enzyme inhibitor, they will expect you to understand that this is an inhibitor which could inhibit reverse transcriptase, therefore inhibit the virus from being able to turn that RNA into complementary DNA, or it could be an enzyme inhibitor that's going to inhibit an enzyme that allows for this cDNA to be integrated into the DNA of the host cell. So if you understand what they're actually asking when they give you information, they're assessing, do you actually know how enzyme inhibitors work? And if they tell you that it is a non-competitive inhibitor, do you know that that would bind to an allosteric site, therefore changing the tertiary structure of the enzyme, making it unable to function? So if they're asking questions on this, they will be looking at you applying your knowledge to whatever concept they send in the question. Now, another one you need to know about is the influenza. So the influenza obviously is one that affects animals. Now, it affects the ciliated epithelial cells and it kills them. So this is where they could apply this to your exchange surfaces topic area. Do you know the role of ciliated epithelial cells? So if the influenza virus affects the ciliated epithelial cells, that therefore is going to stop the ability of clearing out any of the trapped pathogens that have got into our airways. Now, not only does the influenza virus cause this, it also causes an increased release of cytokines. So because we have an increased release of cytokines, this causes inflammation. So remember, cytokines are a chemical messenger. Inflammation is where you have swelling in the area. So you've got an excess of tissue fluid. So those fenestrations have become wider. There's more leakage. We are flooding the area. And also it ends up with an increased mucus production. So we've got ciliated epithelial cells not functioning and the goblet cells are producing an excess of mucus. So this can become a real problem. So that loss of the ciliated cells reduces the removal of this mucus, therefore making the airways more vulnerable to other infections like bacterial infections. which is why it can result in pneumonia, which is a bacterial infection, because someone started off with the influenza virus affecting their cells. Now, what we tend to do when we are suffering with the flu is we tend to cough a lot. Well, if we're coughing a lot, what we're trying to do is we're trying to move that excess mucus back up and out of our airways. So we're trying to clear our airways. So we tend to cough. And obviously with the increased mucus production, that ends up with a lot of congestion. So the important thing to note, again, this is where they could link it, is multipotent stem cells in the epithelium. Multipotent, they are stem cells that have the capacity to divide and differentiate, can produce new ciliated epithelial and goblet cells to replace those that are damaged. But it takes time, which is why you can suffer with the flu for a longer period of time, even though your symptoms seem to be lesser than they were originally, because it takes time to repair the damage that has been done. So this is where, again, they will do this interlinking. So transmission then, if someone's coughing or sneezing, they could transmit that virus in the droplets that are coming out of their body as they cough and as they sneeze. So droplet infection. And if those droplets land on a surface which someone else then touches and then moves to an opening in their mouth, that can then transmit it on. So how do you treat it? Well, you need to rest. You need to rest because you need to allow your immune system, your specific immune response and your non-specific immune response to come along and to fight the infection and to repair the damaged cells. So it is important that you rest when you are suffering with a flu infection. Now, another one is to do with our plants. So the tobacco mosaic virus is the one you need to know about when it comes to plants. So this is where you have a single-stranded RNA virus, which acts like mRNA. So if, again, they're going to give you a question, they may give you a bit of information and then expect you to apply your knowledge. So if you've got a single-stranded RNA, which acts like mRNA, what you understand then is that that can be translated to produce the proteins. If that single-stranded RNA binds to the ribosomes, that will then code for the sequence of amino acids. So again, it's the interlinking that you need to think about. So the cell is going to produce viral proteins. Now, what tends to happen if there is a plant which is infected with the tobacco mosaic virus is there's going to be damages to the leaves. Now, this discoloration, lack of pigment, is why it's called the tobacco mosaic virus, because of that mosaic effect it will have on the leaves. So lack of pigment, lack of photosynthesis. If there's no photosynthetic pigments to absorb the wavelengths of light, therefore there'll be no light dependent reaction for those of you in year 13. If you've not got any light dependent reaction, you won't have any light independent reaction. That will therefore cause a lack of necessary nutrients for growth, and therefore you'll end up with stunted growth and reduced yield in your tobacco mosaic plant that's been affected. Now, it is important for you to note, if you're currently year 12 and you're sitting the AS exam, you will only be asked questions on the fact of it being damaged leaves, stunted growth, reduced yield. If you are year 13 and you are sitting the full A level, they will expect that interlinking knowledge of how that damaged leaves, that discoloration, could then link to photosynthesis. So this is module 4 and photosynthesis is module 5. So this would be a question that would come up in your paper 3, for example. So how is it transmitted? Well, it's actually transmitted through direct contact. So if there is a wound in the plant, it will allow entry into the plant, and then that will allow for that to move between the cells via the plasmodesmata, and it can spread through the plant, through the vascular system. So that direct contact from one that has been infected to another plant, if there's any type of wound, that can then end up with it spreading quite quickly. or even if there is a close plant close by where the wind causes the leaves of an infected plant to touch the leaves of an unaffected plant, that as well could cause transmission. So what's the treatment? There is no cure. You need to remove and you need to burn the infected plant and also disinfect the tools that have been used to reduce the spread of the virus. So it is important that you note the difference between the pathogen type and the diseases, the communicable diseases that you need to know about. But a lot of the time it will all be to do with the interlinking topic areas. So let's have a look at the bacterial pathogens then. So bacterial pathogens that you will be asked about, the key thing to note is that there's lots of different shapes. So they could be rod, they could be spherical, they could be comma shaped, they could be spiral, they could be corkscrew. Now, this is a way in which we can identify which type of bacterial pathogen we have based on the shape of the bacteria. Now, it's important to note that these bacteria are very small. So how do we identify which one we're looking at? Well, we tend to look at their wall structure and we use the staining process to help us. So if it is a gram-positive bacteria, for example, MRSA, they have a thick peptidoglycan wall and they will therefore colour purple or blue when we use a crystal violet stain. If they are gram-negative bacteria like E. coli, they have a thin wall, still peptidoglycan, and they will stain a different colour, which will be pink, and they will use a different stain. Now, you're not expected to memorise the stains, but you are expected to understand what it means if they are using different coloured stains. So it's important to note that this could be testing your understanding on differential staining. So they will give you information and then expect you to apply that to the information in front of you. So the ones you do need to know are tuberculosis. Now, tuberculosis is a slow-growing pathogen and it has a thick cell wall. So it survives, interestingly, inside macrophages. So basically it hides from the immune system by surviving inside your macrophages. So therefore, the immune system actually is really fascinating, and what it does is it tries to contain the infection by surrounding the infected macrophage. But what tends to happen is if this breaks down, it can actually destroy lung tissue and then spread to other tissues. So it is a slow growing pathogen, but it can spread if it is active. So the important thing for you to note when it comes to TB is how it's transmitted. So the transmission is through inhalation of infected droplets. Now, there's coughing, sneezing, or talking could obviously result in the pathogen being transmitted. There is a lot of issue with regards to the TB pathogen actually being transmitted with animals, and one of the ways is the fact that the badgers could actually transmit it with infected droplets to cattle. Now, what they tend to do is things like culling of the transmission organism. Now, that can become a problem because obviously you have to think about ethics. So the treatment, if someone does have tuberculosis, would be giving antibiotics or a vaccination as a preventative. So again, this is a way in which they could be assessing your knowledge on antibiotics and how we should give them and use them, or vaccinations and how the vaccination would result in the active immune system being useful in preventing the spread of TB. So you could potentially have a way in which you kill a organism that potentially transmits the infection, but there's ethics involved in that. We could treat it with antibiotics, but remember, we may result in antibiotic resistance, so a way in which they could go down the topic area, or we could vaccinate against it as a preventative. So another one you need to know about is ring rot, and this one is in the plants. So this is potatoes, tomatoes. This is caused by a gram-positive bacteria, and it damages the leaves, the tubers, and the fruit. So if they're going to ask a question about this, they're looking for you to understand how is it transmitted. Well, it can be transmitted in infected seeds. It can be transmitted in contaminated tools. It can be transmitted in water. And it can also be transmitted through plant debris. So if we're going to treat it, there is no cure. What the farmer must do is they must remove the infected plants, remove all parts of the infected parts of the plant, and they must wait 2 years before they can grow the potatoes again. And that is because of contamination in the soil. So it's important to note that there are potentially some time periods that you need to wait. So if they are going to ask a question about this, it will be, as I've said before, them giving you some information and then expecting you to pull out from that what they could do to prevent it. So another type of pathogen is a Protista pathogen. Now, Protista, again, is a eukaryotic organism. So they may ask about the fact that it's a eukaryotic organism. You should know that it has a nucleus, that its DNA is associated with histone proteins, that it has membrane-bound organelles. So if they're going to ask questions, they could look at cell structure, as well as looking at you being able to identify it as a pathogen. Now, with the Protista, they actually use people or animals as hosts. They're the ones that are going to digest and use the cell components to reproduce, and they may need a vector to transfer them. So it's important to note that the malarial parasite is one that you need to know. So this is Plasmodium. The malarial parasite reproduces inside a mosquito vector. And it's an interesting one because it needs the female mosquito, the Anopheles mosquito, to be able to reproduce. So when the mosquito feeds, if it has been infected, it will actually transfer the malarial parasite into the human bloodstream when it feeds on the hemoglobin, and therefore it will rupture the erythrocytes. So the mosquito is going to feed on the human blood. As it feeds, when it actually pushes its mouth part, the proboscis, into the skin, what it does is it releases saliva to numb the area. So you don't actually feel the mosquito as it's biting. What's actually happening is when it is putting the saliva to numb the area in around where it's going to feed, what happens is the malarial parasite is going to go into the bloodstream with the saliva. So that is now in the bloodstream, and that malarial parasite is now going to feed on the hemoglobin. Hemoglobin, remember, is a protein with the heme group attached to it, so it has a prosthetic group. So it could be where they bring in biological molecules and look at the protein structure in this topic area. It will then rupture your erythrocytes. So if it's going to rupture your erythrocytes and it has declined your, reduced your hemoglobin levels, what's going to happen is you're not going to be able to transport oxygen as efficiently, which becomes a problem. So how is it transmitted? It's transmitted through that vector. So the vector is the female mosquito, and therefore the treatment is actually, there's no cure. It's important to prevent it. So the prevention is important through controlling that vector. So if they're going to ask a question, they'll be looking at you saying things like using mosquito nets, protective clothing, could be using insect sprays like insecticides. However, using insecticides could have an effect on the biodiversity. So that is where they could link in your biodiversity topic area. So it is important that you note how that preventative measure is a positive measure, but if they're asking you to evaluate, it could also be a negative measure. So another Protista pathogen is late blight. So late blight is a problem when it comes to potatoes, tomatoes, for example. Now, this is because there are spores which will land on the leaves, the stems, or the tubers. So the Protista pathogen releases spores as it goes through its reproductive life cycle, and those spores will land on those leaves, stems, and tubers. If the conditions are right, those spores will then germinate. What that means is they will begin to grow. How they do that is they're going to release enzymes which are going to break down the plant cell walls, and then they can absorb the plant's nutrients. So what they're doing is they're releasing enzymes which are going to hydrolyze the cellulose which is in the plant cell wall, and then they can then use the plant's nutrients to build, assimilate, and make more of what they require. So how are they transmitted? Well, the Protista pathogen produces these many spores that can be spread through the wind, they can be spread through the rain, so through water, and they can be in contaminated soils. So the treatment is no cure, but careful removal of all parts of an infected crop. So that means that you need to make sure that the soil is not contaminated as well. And then the last one you need to know about is the fungal pathogen. And these are ones which are going to digest living cells extracellularly. So again, releasing enzymes which will then digest down, and then they can absorb the nutrients. So they've reproduced by producing, again, millions of tiny spores. So just like the Protista, they also produce spores, and those spores can be transported over large distances. So this is why some fungal infections and some Protista infections can result in being infected in different lands because of this ability to transport over large distances. So that is a way in which they will look for that application by telling you of a isolated island which now has an infection. Where did that communicable disease come from? If it was fungal or a Protista, then it's to do with the transport of the spores. Now, the two that you need to know is in plants, the black cicotoca, which is one that affects bananas. So what you have is the fungal hyphae, the extensions, actually penetrate and digest the cells, turning the leaves black. Again, if we're going to affect the leaves, if the leaves are being affected, that therefore means that they're not going to have the photosynthetic pigments. You've not got as much surface area for absorbing the light, less photosynthesis, less growth. How do we treat it? Well, it's a fungal pathogen, so therefore we would use fungicide. Or we could potentially genetically modify them to become resistant strains from the pathogen which is affecting them. So again, it could be asked in many different ways depending on what they want to assess in the question. And then we have athlete's foot, which is the other one you need to know with regards to humans. This is where they're going to digest because they're in a warm, moist area. So the skin between your toes is a nice, warm, and if you're wearing socks and you get hot, you produce a lot of sweat. It's very moist. Therefore, they've got the right conditions for digesting that area. And what that can do is that can actually result in them breaking the skins and causing sores. How's it transmitted? Well, it's through direct contact. If somebody has athlete's foot and they walk around barefoot, and then someone else who doesn't have the infection then walks on that same surface, that direct contact can result in that transmission of the fungal pathogen. How do we treat it? Well, we use fungicide creams. So there are lots of different ways in which animals and plants can have this transmission, whether it's direct contact, inoculation, ingestion, fomites, if it's droplet infection, if it's vectors. with plants, direct contact, that soil contamination, and vector. If they're asking any specific question, they'll have given you the information in the stem of the question. They want you to then apply your knowledge. But there are specific factors that are going to affect that transmission of the pathogen, that are going to increase the rate at which it's transmitted. And one of those is living conditions when we look at animals. Overcrowding. If they're asking about areas, is there lots of people living in one household? What's the hygiene like? Are they able to isolate away, or are they in close proximity? Any of these are going to increase that transmission. If they're having poor nutrition, they're therefore going to have a compromised immune system. They're not going to be able to fight the infection fast enough. That could result in an increase in the transmission. Also, if they are taking, for example, immune suppressant drugs, or if they have HIV, they are going to be immune compromised, and that can also have an effect. Social factors. What is their income like? For example, are they able to pay for healthcare? Do they have access to it? Can they travel to somewhere where they're able to get that care, or are they able to travel somewhere where they may get infected? So for example, when I went traveling around Southeast Asia, there was chance of me being affected by communicable diseases which I would never have experienced in the UK. Therefore, I had to be vaccinated against them. I therefore had to pay privately for that. Is that possible? So what's the social factors? Can the people be vaccinated? Is their occupation going to result in an increase in the risk factor? The other side of things is socioeconomic factors. Things like, are people trained? Are there trained health workers that can help? Or is there the right information to warn the public of the risks of these diseases? So whenever they ask questions on this, they will give you a information, and then they will expect you to apply knowledge. So there'll be some bits of information about healthcare or trained workers, or they won't say anything. So then they'll expect you to state things like improve the training of healthcare workers to stop the transmission. Make sure, for example, that they sterilize all their equipment, or make sure they are warned against the spread. For example, HIV, unprotected sex, sharing of needles, warning the public by making it educational, that they are aware of the risk, could prevent it from being transmitted. And also the climate is something to factor in. When you change the climate, for example, if the temperature changes, or if there is more or less rainfall, that could affect the presence of vectors, or it could affect the presence of pathogens because you have more favorable conditions. So have they given you any information on the temperature or rainfall? And therefore, if there are vectors, if there's more rainfall, therefore the spores can be transmitted further with more rainfall. Or if the temperature changes, does that mean that there's now a vector in an area that wasn't present before? So I hope you've liked this video, and if you have, then please do click on the like button and subscribe to my channel. Also, if you haven't done so already, do check out my revision platform, www.aiqchat.com.